Hello, my name is Professor Hudson, and you're about to view narrated PowerPoint lectures for Chapter 11. In this last segment, we will look at how human genetic disorders are inherited. And also, we will begin discussing a little bit about inheritance patterns that do not follow traditional Mendelian genetics. We will discuss that more, though, in Module 7, Part 2. So, just like body traits can be inherited through genetic rules, genetic disorders can be inherited by similar rules. Genetic disorders are medical conditions caused by alleles inherited from parents. These disorders can be autosomal recessive or autosomal dominant. An autosome, remember, is any chromosome other than the sex chromosomes. Oftentimes, in order to understand how genetic disorders are inherited, pedigree analysis is done. This is because you cannot do test crosses and practice matings with humans. In a pedigree analysis, inherited traits are analyzed over the course of a few generations in one family. This slide shows an example of a pedigree for a dominant trait and for a recessive trait. It also shows the shorthand that you use when reading pedigrees. Note that oftentimes the Roman numerals are used to designate the generations. A square is used to represent a male, and a circle is used to represent a female. A square and a circle with a line between them indicate the parents. And then the offspring are written beneath. Here is an autosomal dominant pedigree chart. Some human genetic disorders are caused by dominant alleles. A dominant disease can be transmitted to offspring if at least one parent suffers from the disease and lives long enough to reproduce. Dominant disease alleles also came about from mutations in the sperm or egg of unaffected people. Some dominant alleles encode an abnormal protein that interferes with normal function, or some dominant alleles encode proteins that are toxic. An allele may be dominant if it encodes a protein that is overactive or that is active at inappropriate times. Note that in an autosomal dominant pedigree, usually affected children will have at least one parent that also has the disease. In the case of autosomal dominant, you do not need 2 copies to have the disorder. You only need 1 copy. This means that the heterozygotes are not considered carriers, but they actually have the disease. 2 unaffected parents can produce an unaffected child. And 2 unaffected parents will have children that are normal. Both males and females are affected with equal frequency. Huntington's disease is caused by a defective protein that kills cells in certain regions of the brain. It is a dominant disorder that causes a slow progressive deterioration of parts of the brain. This disease results in a loss of coordination, failing movements, personality disturbances, and even death. This is an autosomal dominant disorder. The disease usually does not show up until adulthood, which means most of the time people have already had their children before they realize they had it. Some other examples of autosomal dominant disorders are osteogenesis imperfecta, also called the brittle bone disease, neurofibromatosis, where you get small benign tumors all over your body, and in a situation sometimes where children are born with a little extra finger on one hand, that is also an autosomal dominant disorder. It is also an example of incomplete penetrance, which we'll talk about later, in that you could be dominant for the mutation that causes the extra digit, but you may not necessarily actually have been born with the extra digit. This autosomal dominant mutation shows up sometime, but not all the time. The African American lady on the left had the autosomal dominant for the extra digit. In her case, it probably was not removed when she was born, which is why it is still there. The Caucasian in the right side of this picture is just included for contrast in that this person probably has some other disorder. The autosomal dominant feature we're talking about with the extra digit in this particular case is given by the lady with the African American features here. And this is usually an isolated event that is not related to any other disease or disorder. When doing Punnett squares for autosomal dominant disorders, you use the same setup rules for the Punnett square. However, the way in which you read the phenotypes is a little bit different. Set up the Punnett square here and see if you can answer the question. We will discuss how you solve this in class. Huntington's disease is caused by the allele H, which is completely dominant to the normal recessive allele H. Consider a couple where the man has the genotype of heterozygous and the woman has the genotype of homozygous recessive. See, can you figure out what the chances are that the first child will develop Huntington's disease? Try to work this on your own and we will answer it in class. With autosomal recessive pedigrees, it's the opposite situation. In those cases, once again, you have alleles produced by mutation that code for non-functional proteins. These non-functional proteins are recessive to the ones that code for functional proteins. The presence of one normal allele may generate enough functional protein to enable the heterozygote to be phenotypically indistinguishable from homozygous with two normal alleles. In the case of autosomal recessive inheritance, the heterozygotes are considered carriers. Usually they appear normal, however they are carrying the mutation in their gene pool. Recessive genes are more likely to occur in the homozygous combination when related individuals have children. With autosomal recessive inheritance, most affected children have an unaffected parent, the heterozygotes have an unaffected phenotype, two affected parents will always have affected children, and the close relatives who reproduce are more likely to have affected children. Both males and females are affected with equal frequency. Here are some examples of autosomal recessive disorders. Albinism, or whether or not a person is born an albino, results from a defect in melanin production. These people inherit the gene for melanin production, but an enzyme necessary for processing this gene and protein causes them to not produce any melanin. Melanin is the dark pigment that colors skin cells. Melanin is produced by the enzyme tyronase. An allele known as TYR for tyronase encodes a defective tyronase protein in skin cells. So even though these individuals have the gene for producing melanin, they actually do not produce any melanin because this enzyme is not functional. Humans and other mammals who are homozygous for the TYR have no color in their skin, fur, or eyes. The skin and hair will appear white, and their eyes will appear pink. Sickle cell anemia is another example of an autosomal recessive disorder. Sickle cell anemia is caused by a defective allele for the hemoglobin protein. People homozygous for the sickle cell allele synthesize only defective hemoglobin, and therefore they produce mostly sickle cells. Although heterozygotes have about half normal and half abnormal hemoglobin, they usually have few sickled cells and are not seriously affected. Because only people who are homozygous for the sickle cell allele usually show symptoms, sickle cell anemia is considered a recessive disorder. About 5 to 25% of sub-Saharan Africans and 8% of African Americans are heterozygous for the sickle cell anemia. But the allele is very rare in Caucasians. The large proportion of heterozygotes in Africa exists because if you're heterozygous for the sickle cell anemia trait allele, you have some resistance to infection by the parasite that causes malaria. The rarity of heterozygotes in Caucasians corresponds with the rarity of malaria in northern climates where many people of Anglo-Saxon Caucasian descent originated from. So therefore, in these northern climates, where malaria is not really an issue, the immunity provided by being a heterozygous has no selective advantage. Autosomal recessive cystic fibrosis. This is another example. With cystic fibrosis, there's a defect in a channel protein that allows chlorine ions to move across cells. This upsets the water balance in that osmosis does not occur correctly. People with this homozygous recessive double mutation will produce extremely thick mucus in their organs and digestive tract and lungs. Here's another practice question you can try. If the parents are heterozygous for cystic fibrosis, then each offspring has what chance of having cystic fibrosis? See, can you set up the Punnett square and work the problem to solve this? We will discuss it in class. Let's conclude by talking about genetic disorders that are caused by an abnormal number of chromosomes. In this situation, the mutations are not within the chromosome, but actually there are defects in the number or size or shape of the chromosomes. Nondisjunction is when chromosomes separate improperly or when chromatids separate improperly during meiosis. This can cause the sex cells to have too many or too few chromosomes. Most embryos that arise from fusion of gametes with abnormal chromosome numbers will spontaneously abort. However, some will survive to birth and beyond. Trisomy 21, also known as Down syndrome, these individuals have 3 copies of chromosome 21. So either the egg or the sperm did not undergo meiosis correctly. It occurs in 1 out of 800 births. Down syndrome includes several distinctive physical characteristics, including weak muscle tone, a small mouth, and a large tongue, and distinctively shaped eyes. Down syndrome is characterized by low resistance to infections, about half have heart defects, and varying degrees of mental retardation, often severe. Just like you can have abnormal numbers in the autosomes, you can also have abnormal numbers of sex chromosomes. This slide shows 2 examples of disorders that are caused by abnormal number of sex chromosomes. In this situation on the left, we have a woman that has Turner syndrome. During meiosis, at some point, either the egg or the sperm, most likely in this case, it could be either the egg or the sperm, some did not have the right number of chromosomes, and so she ended up missing an X chromosome at fertilization. Women with Turner syndrome, they're still considered women even though they have only one X chromosome because there's no Y chromosome. At puberty, the deficiencies prevent these females from menstruating or developing sexual secondary characteristics. Hormone treatment can promote physical development, but the affected women usually do not produce any eggs and will be infertile. Some additional symptoms of Turner syndrome are short stature, folds of skin around the neck, and increased risk for cardiovascular disease, kidney defects, and hearing loss. Because they only have one X chromosome, women with Turner syndrome are more susceptible to recessive disorders such as red-green color blindness or hemophilia. The male on the right has Klinefelter syndrome. In this situation, he has too many X chromosomes. He can be XXY or XXXY. Most show no symptoms other than some partial breast development, broad hips, and small testes. Usually, though, they are infertile, and that's usually when the diagnosis for Klinefelter syndrome is made. This slide shows some examples of nondisjunction in the autosomes and sex chromosomes and the frequency with which they occur. This concludes our narrated PowerPoint lecture for Module 7, Part 1 on genetic inheritance.